So my presentation was talking about the fact that nowadays the current treatments haven't really evolved since the last three decades, causing the problem that a lot of patients that are either refractory to the treatment or are relapsing have absolutely no solutions. So my goal was to find new therapeutic targets, and the best way to find new therapeutic targets is to interrogate the genome by performing genome-wide CRISPR screens. So with this technique, you can actually interrogate genome and turn off one by one the different genes of the cancer cells, and you in the end find out which genes are essential for the cancerous cell to continue to live or to proliferate. So from there, I found different types of genes, try to find genes that would be like very targeted for a specific type of leukemia. So in this case, TLLs that are driven by Notch. So from there, I tried to compare to other types of leukemias and also normal cells to see if by turning off these specific genes, I was only harming, let's say, the Notch-driven cell lines or Notch-driven cells compared to normal cells. And then you then limit your number of genes. And from there, the interesting bit is not to satisfy yourself by only showing, let's say, this data. You try to explain why this cancerous cell needs this specific gene or this specific pathway to be able to continue to progress. So that's how I found NADK, and in fact a lot of different genes that are implicated in the mitochondrial, let's say, mechanism, metabolism. And used a drug that was commercially available and starting to show that it has a very specific role in the ability of Notch-driven cells to be relying on these genes or on these pathways to be able to proliferate. So the drug that I used was thionicotinamide. So unfortunately, the potency of this drug is not that great, but it does show a mild effect. So there's always a possibility to try to improve this initial molecule to make it more efficient, or use it with other known therapies that would be making the treatment more efficient. So this hasn't really been shown yet. What has already been shown, and what I proved also in my talk, is that when you initiate the response by Notch, it does, or if Notch is overexpressed or activated or overactivated, you have a reduction of the ROS levels in the cells. And if you block Notch, you then have an increase of ROS levels into the cell. And the role of NADK is to produce the metabolites that will be used to detoxify ROS. So it seems there is, in a way, a connection, a correlation between the activation of Notch, NADK, and the reduction of ROS levels in the cells. For the moment, I only observed in TLLs, but it's true that Notch has a role in breast cancer and other types of cancers. And the problem with Notch is that it has a kind of ambivalent role. So in certain cancers, the overexpression of Notch is beneficial for the cancer cells, and other cancers, the repression of the receptor Notch is going to be beneficial for the cancer cells. So it has, for example, in esophageal cancer, the repression of Notch is actually what drives the esophageal cancer to progress. So the next steps is try to understand why Notch is so dependent on having ROS levels so low. Is it because they are using a specific pathway that produces more ROS than other cell lines or other types of cancers? And try to see how it fits all in the metabolism and what has already been shown. So Adolfo Ferrando showed that the glutamine pathway is very important for Notch-driven cell lines. Haifantis showed that the heat shock proteins are also important for Notch-driven cell lines. So try to see how what I found is able to fit between these two stories.